Thanks very much for the opportunity to present today. I'm going to be talking about Sernova's technologies and our advancements as we're going forward. Please be aware of the forward-looking statements. So what is Sernova about? So Sernova is a cell therapeutics company, and we have a cell therapeutics platform that is able to not just help patients to improve their disease, but actually to provide a functional cure. So we have a number of different indications, and the question is, how are we doing that? And we have something that we call the cell pouch system. And this involves a small medical device that I will talk about that is placed deep under the skin in the abdominal area. And it is very porous. It fills in with highly vascularized tissue. It forms an organ-like environment for the placement of therapeutic cells. And the second part of this are the cells themselves, and we're focused right now on a clinical trial with donor islets for our phase 2 clinical trial for type 1 diabetes, but we are also working on an iPSC-derived technology. In terms of our other programs, we have additional programs in hemophilia A, which is an ex vivo gene therapy approach that I'll talk about in a few minutes. And we are also the first company in the world to be working on treating thyroid disease. So in terms of our advancements over the past little while, we've been working on a phase 2 clinical trial at the University of Chicago and with type 1 diabetes. And we currently have 5 out of 5 patients that have become insulin independent. And the longest patient is insulin independent for 3.5 years. So what's really important here is, from the device perspective, virtually all companies that have been working on devices for cell therapy have shown poor results and a lack of being able to get the appropriate amount of insulin into the bloodstream and then fibrosis of their device. So the fact that we have patients that are insulin independent for over 3.5 years is showing that we have a device that is actually forming that organ-like environment and working appropriately. In May 2022, and I mentioned that we're working with donor islets that has a limited supply of cells, we did a deal with Evotec. And the idea here is that we want to work with iPSC cell clusters. We had a number of collaborations, and we found that Evotec was the best partner for us, and we'll talk about that in a few minutes. We have also a collaboration with AstraZeneca, and that collaboration is exploring other cell therapy approaches that we can use with our combined technology. So we're working on these several different programs, but we're also now looking at a whole series of different opportunities. And the approach that we're taking is that we're evaluating a scientific approach, and then looking at patient impact studies and figuring out what the right implications or right indications are going to be. We're then doing internal development up to a phase 2 program, and then looking at licensing out that technology. So we're going to be able to build value for multiple indications as we're going forward. So to talk a little bit more about our cell pouch technology, what we found is that we work very closely to really understand how devices and cells could be placed into the body and what the body really needs from a biological perspective. So what we learned is that if we develop a device that is very, very porous, the body will naturally treat that like a scaffold, very similar to an organ. And what it typically will do is fill in all the pores with highly vascularized tissue, and we have these small space-holding plugs within each chamber of the device. And after about 3 weeks, we get these beautiful, highly vascularized tissue chambers, pull out the plugs, and then we can put in the therapeutic cells, and then the tissue matrix grows around the therapeutic cells, and then those cells survive and start to produce their product. What is really important and differentiates Sernova from other companies is that we place the device in the body first, allow it to form that organ-like environment, and then put the therapeutic cells in. Our competitors are typically putting their cells into the device and then putting the device in the body. And it's a very difficult situation to keep those cells alive. So in terms of the process that we're going through, with a very disruptive technology, what we've been doing is first working with the donor islets to prove that the device is working. And obviously, islets are from your own body, we already know that they work. So with all of those data, then we're moving forward with our iPSC technology and using the immunosuppressive medications as our first foray. And then for our third approach that we're doing, we're working on locally immune-protected cells within the device itself. So all three of these programs are moving forward at the same time. So if we talk about the market, first of all, when you think about type 1 diabetes, the cost of type 1 diabetes is not the cost of insulin. It's really the cost of the medical issues around the side effects that occur with type 1 diabetes. So we have done payer studies to be able to understand this, and they have come back and said that with a product that lasts approximately 2 years, and we're obviously already beyond that with the 3.5-year success rate, they would pay approximately $300,000 per patient. And if you look at that, the first group of patients that we're looking at treating are the most severe diabetic patients. These are people that cannot produce insulin on their own, and they have severe hypoglycemia unawareness. And if you think about hypoglycemia unawareness, these are the patients that when they take an insulin injection, their blood sugar levels can drop to dangerous levels and they can go into a coma. So that's about 400,000 patients in the US alone, and 1% of that represents about a $1 billion market. When we go to the broader market, 1% of the broader market, when we use our immune-protected, local immune-protected device, then that's about $5 billion for 1% of the market. So it's very, very significant from that perspective. If you look at our pipeline, again, we're starting off with working on the donor islets. It's a very, very de-risked approach, and that has really taught us a lot about what we're going to be doing going forward with our stem cell-derived technologies. From the hemophilia A perspective, we have been working with blood samples from hemophilia A patients. We're able to isolate the endothelial cells, correct the gene for factor VIII, scale up those cells, put those into cell pouch, and we've been able to show really, really good efficacy in animal models by releasing factor VIII into the bloodstream. So this is an ex vivo gene therapy program where we can put the cells, not infecting the cells of the liver, but actually putting those cells into the device itself. And our first approach is going to be an autograft approach. And then as we're going forward, we're looking at an iPSC-derived technology. And the third approach that we're taking is with thyroid disease. And here, similar to our other approaches, we're taking patients that have had their thyroid gland completely removed for benign purposes, and we're able to take the healthy tissue and place that into the device. And in our preclinical studies, we've been able to show that we can completely recover the feedback loop between the brain and the cell pouch thyroid tissue. So this is something that we're really excited about moving forward. So in terms of our phase 2 study, we are working for diabetes, we're working with patients, again, that are the sickest of the sickest patients. So these people have no ability to produce insulin in the glucose tolerance test. And then they also are, not only are they not able to produce insulin, but they also have no islets function whatsoever. So as we're going forward with these patients, when we put the cell pouch and the islets into the device, when we start seeing insulin in the bloodstream measured by C-peptide, which is a biomarker for insulin, then we know that that insulin is coming from the cell pouch. We have 2 cohorts in this study. The first cohort really was a dosing study to teach us what dose and dose density was required. And with our 5 insulin-independent patients, what we actually learned is what dose is required to get insulin independence, we believe, in all of the patients. So we're now moving forward with a second cohort of patients. And that cohort uses a device that's a little bit larger, provides 50% more room, and we can spread the islets out so they have a much better breathing space. And we now have a very good idea of what is required to be able to get insulin independence in terms of the dose. And interestingly, in our iPSC technology, to bring that back in relationship to the donor islets, we've been able to show that we're projecting that the exact same dose can be used for getting insulin independence. So again, we just presented data at the recent American Diabetes Association meeting, and our safety for our device and our therapeutic cells is really, really positive. Again, we also have sentinel devices that come out at the 90-day time point, and we do extensive histology to show that those islets are surviving, they're producing insulin, and that they're highly vascularized. So we've seen this very, very consistently in all of our patients. So the second cohort, which has the larger devices, we now have 6 patients that have been enrolled, and that's 6 of the 7. And we have now put islets into over 4 patients. And the first evaluable patient is showing good levels of C-peptide and showing both in fasting glucose levels and also a glucose tolerance test. So it's looking like the second cohort of patients with the larger devices is working extremely well. So we're really thrilled about that. So as I mentioned, we did an analysis of a number of different iPSC technologies and had a number of collaborations with pharma companies. And we ended up choosing Evotec, and Evotec chose us because we believe they have the best iPSC technology out there. And this agreement was announced in 2022. The way it's working is that Sernova and Evotec are funding the project up to the IND stage together. And then after that, Sernova will take over the clinical development approach. Also, Evotec is responsible for producing the GMP iPSC cells. So they have purchased a facility to be able to do that. And then they're also responsible for the commercial manufacturing. So what's really good is that Evotec is expert in this particular field, and it allows us to be able to get very, very high quality cells that then can go into our device. And at this point in time, we're working on the scale-up activities to get to the clinical trial and do our final toxicology studies in preparation for going into the clinic. So one of the questions you can ask with the cell therapy approach is, do you have a commercial approach? And what we really liked about the cells from Evotec is, first of all, that we have a differentiation protocol that brings those islets up to the point right before they're producing insulin. So once the cell starts to produce insulin, then it has a very high metabolic rate and is very unstable. So by bringing the cells up right before that, then when they go into our device, they can spend their time in grafting, and then a couple weeks later, they start to produce insulin. So we feel that this is the most ideal way of doing it. The other thing, unlike our competitors, is that we're able to get islet clusters made, and we found a method to be able to cryopreserve those islets. So therefore, we can control the islets in terms of time and space, so that if you think about a patient that's getting treated, they would go to their endocrinologist, they would get the cell pouches placed in. About 3 weeks later, they would order the cells, they would go right into the device, and then the patient would titrate down the insulin as the cell pouches islets were taking over. So we've done extensive studies with Evotec. They've done studies up to a year with their iPSC cells. So one of the big questions is, are these cells stable? Do they last a long time? And these are very, very robust and consistent. The other thing is when we've supposed to with their cells in the cell pouch, and we've shown that we're able to get down to the set point of humans in terms of the blood sugar control. And what's really, really important in the de-risking of this technology is that we've compared the production of C-peptide and also glucose tolerance tests to human donor islets, and we find that they work exactly the same. So we're now able to take all the data from our phase 2 study for donor islets and use that as we're advancing our iPSC technology into the clinic. So we feel like we've worked really hard to de-risk the iPSC technology going forward. So the next question you can ask is the immunosuppression. So we have worked and significantly improved the immunosuppression protocols for our current patients. We've been able to reduce the tacrolimus significantly by using an additional drug that we've been working with that is very, very well tolerated in the patients. So that's a really good improvement, but at the same time, we want to have local immune protection. And we have a technology that we've been developing that is a polymer, cross-linked polymer technology that goes around the islets, and then those cells then go into the cell pouch chambers. So again, we're able to get normal blood sugar control without any reduction in kinetics. and we can also get immunoprotection. So this technology is advancing forward to the clinic. So we have a number of important milestones that we've already come out with this year, but we're going to be providing a lot more information on our phase 2 clinical trial, the second cohort by the end of this year. We have more information on the thyroid program and hemophilia going forward. And as we're moving into 2024, we're going to have a lot more information around the manufacturing process and processes moving into the clinic with our iPSC technology. So I just want to mention our management team. We have a very, very strong management team with at least 30 years experience in each of us. And I have been CEO since 2009, and I've just turned over the CEO role to Cynthia Pusanen, who I'm very happy to have on board right now. And I've transitioned to the CTO to be able to develop the next technologies for the company. So thank you very much.